Good morning, everybody. Welcome to VSPG. Today we have a presentation by Dr. Andreas Kappler from the University of Tuebingen, Geomicrobiological processes in banded iron formation, deposition, banding, and mineral diagenesis. Let me find, next week we have a talk by Rosalie Tosteven. And I have that title here. Searching for the signals of oxygen in microbial reefs. Rosalie's at the University of Cape Town. So join for that. That sounds like it's going to be particularly exciting for geochemists, sedimentary geochemists. So today, Andre is going to go ahead and introduce Andreas for us. Go ahead.
Yeah, it's a pleasure to introduce Andreas Kappler. Andreas is originally from Germany. He did his undergraduate degree in chemistry, and when he moved for his PhD to the field of environmental microbiology, both degrees he did at University of Constance in Germany. He followed with a 2-year postdoctoral position in ETH in Zurich, working on environmental chemistry. And when he moved to Caltech, where he worked with Diane Newman in geobiology, and that's, I suspect, where his interest in ion cycling in ancient and modern environments evolved. And in 2004, he took position at the University of Tuebingen, where he is based now, and he lead a research group in geomicrobiology. And he done a lot of work on sort of modern and ancient microbiology, how it's linked to origin and post-depositional changes in ion formations. And I guess that's what he will be presenting today. So with this, I pass it to Andreas.
Perfect. Thank you very much for the introduction. And of course, thank you very much for inviting me to give this presentation. And as you mentioned, my group is studying ion cycling in modern environments. And we also have this interest in ion cycling in ancient environments. And I did my postdoc, as you said, at Caltech in 2004, almost 20 years ago. And this was when I got interested in banded iron formations and in studying how microbes potentially could have contributed here in the deposition of these rocks that you can see here on the right. Let me start with a very, very brief introduction into banded iron formations. These are sedimentary deposits that contain these characteristic alternating layers of silica and iron minerals that you can see here on the top left photo. They are very old. The oldest ones are up to 3.8 billion years old, down to 1.8 or maybe even 0.8 billion years, related to some glaciations. And the dimensions of some of them are just huge. You see here in the background a photo of the Hammersley BIF, hundreds of meters thick, an area of 1,000 square kilometers. And I got really interested in that because thinking about how these minerals formed in the past, we can potentially learn something about the evolution of the atmosphere. That means the presence of oxygen. They're also about the hydrosphere, the biosphere, what kind of microbes we have had present at that time. And I was in particular interested in the presence of oxygenic photosynthetic organisms. That means the ones that produce oxygen, but also the anoxygenic photosynthetic organisms. These organisms, they don't split water, but they use alternative electron donors, for example, iron 2, and oxidize this iron 2 to iron 3. Now, the interest in these microbes got strengthened by the discussion of different mechanisms, how the minerals that we see nowadays in these rocks could have precipitated partially in combination with the activity of microbes. And a couple of different mechanisms have been suggested that I would like to summarize on this slide and on the next one. For a long time, it was thought that the presence of banded iron formations containing these oxidized minerals is related to the presence of oxygen. And in this first scenario, you can imagine that cyanobacteria produced oxygen And this oxygen was reacting chemically, as you can see here in this equation, with iron 2, oxidizing the iron 2 to iron 3. And if the pH is around neutral, this iron 3 will precipitate as iron 3 oxyhydroxides, potentially forming these oxidized minerals here at the bottom of the ocean. Now, there are also microorganisms that can be active under these conditions and use the oxygen as electron acceptor for an enzymatic oxidation of iron 2. That means you could also imagine the combination of having cyanobacteria producing oxygen, and this oxygen is then used by these so-called microaerophilic iron oxidizing bacteria, which make a living by oxidizing the iron with oxygen and also deposit iron 3 oxyhydroxides in a water column or in a sediment and can precipitate them in such a scenario. Now, there are also two mechanisms that can explain how iron can be oxidized in the absence of oxygen. And one was a mechanism that was suggested already very early, that photooxidation of iron plays a role. In this case, the iron 2 in the water is oxidized with the help of UV light, not catalyzed by microbes, but by a photochemical reaction, also producing iron 3 and hydrogen. In this case, this is where the electrons go. And then the iron 3 also precipitates as iron 3 oxyhydroxides. The question is whether this was really significant, because there were some experiments that showed that if you had silicate present in relevant concentrations of around 1 to 2 millimolar, the precipitation of these iron silicate minerals is too fast, to compete with the oxidation by light. That means it's probably not so relevant for such a scenario. However, there is an alternative, and this is phototrophic iron oxidizing bacteria. This is a group of anoxygenic phototrophs that in this case use iron 2 as an electron source. They use light energy to excite these electrons, and then these excited electrons are used to fix CO2 or bicarbonate into biomass. And these organisms can live in the absence of oxygen. They use the iron 2 that is released from hydrothermal sources, and they live in the photic zone where they get enough light energy. And they could explain how iron 2 is oxidized under anoxic conditions. Now, there's also an alternative mechanism that does not directly include microorganisms. It was also suggested that iron 2 silicates and or iron 2 carbonates can precipitate directly from the water column and become oxidized later during metamorphism. And about this mechanism, I will not talk today a lot. I will mainly talk about the ones just mentioned involving microorganisms. Now, when we think about how this happened in an ancient ocean, this actually doesn't stop at the process of oxidation of the iron. That means in the beginning, you have oxidation of iron by either phototrophic iron oxidation, by these iron oxidizing bacteria, or by a chemical reaction with the oxygen produced by cyanobacteria. But in all these cases, we produce iron 3 minerals that will interact to some extent with the organic compounds that are produced during these microbial processes. That means we have sedimentation of these so-called cell mineral aggregates. And as a consequence, I would expect that at the ocean floor, we have a whole series of microbial processes, including first fermentation, degrading some of the organic carbon. Then this organic carbon that is degraded, including fatty acids and also hydrogen, can fuel microbial iron reduction, producing again iron 2 and bicarbonate. But the fermentation can also go all the way down to methanogenesis, producing methane. And this methane in turn can be oxidized by methanotrophic organisms. These are the ones that oxidize the methane back to CO2, for example, with iron 3 as an electron acceptor. That means you can see that the deposition of the biomass together with the iron minerals initiates a whole series of secondary microbial processes. But even after that, there's another series of abiotic reactions that can happen when we have the deposition of iron 3 and carbon, that means biomass. And over time, we increase the pressure and the temperature, metamorphism takes place. And this can also lead to abiotic iron 3 reduction. That means it's not only, as I mentioned, the oxidation of iron, it's also the microbial processes that result in the sediment. And at one point, it's also the metamorphic processes that can happen later. Now, what I would like to talk today is about the following questions. First, if we consider these cyanobacteria producing oxygen very early, then one of the big questions is, why did it take so long until we had larger concentrations of oxygen accumulating in the atmosphere? Then the second question I would like to discuss with you is the deposition of such huge amounts of iron minerals realistic considering modern phototrophic iron oxidizing bacteria? That means their conditions of living and the oxidation rates. Then one of the fascinating questions is, what caused the bending, as this characteristic stratification, the bending, the alternating bending of iron and silicate minerals, and can we explain this potentially by including microorganisms? And then finally, I would like to show you lab experiments where we studied what kind of minerals do we obtain when we transform these biogenic minerals at higher pressures and temperatures. Now, how can you approach these kind of questions? Of course, you can go into the rock record and can look into the mineralogy, into isotopes, iron isotopes, silicon isotopes. You can also look into trace metal distribution like nickel. You can also look into nutrients like phosphate. You can search for fossils. But what we do is, our lab, we grow microorganisms that we isolated or other people isolated from modern environments, and we study their behavior under conditions that were potentially relevant for ancient oceans. That means we try to simulate these processes that happened on early Earth. Of course, there's this limitation that we know that these microorganisms were isolated from modern habitats, and maybe they behave differently. But this is the best we can do, because we cannot isolate the ones that existed billions of years ago. But we can get some information about how they would have lived under certain conditions, what minerals they would have produced, etc., etc. And this is what I would like to show you today. Let me start with some first experiments that some people in my group did using cyanobacteria. This was a project initiated by Betsy Swanner, who was a postdoc at University of Tübingen in my group and Ronny Schönberg's group a couple of years ago. And she worked together with Markus Maisch, a PhD student. And what they did is, first they assembled this nice column that you can see here on the left that had different sampling ports. And we used glass beads to stratify the column. And we had different inflows and outflows, and we illuminated it and inoculated it with cyanobacteria, with a strain, Synechococcus PCC 7002, that you can see here on the right. And you can see here this layer of green cyanobacteria, and you can also see some layers where you see the orange minerals, the iron oxides. And one of the first observations that we made here is that it looked like that the presence of iron 2 influenced the activity and the growth of these cyanobacteria. That means they didn't really grow quickly everywhere. It looked like it depended on the concentration of iron 2 that was present in the column. Betsy and Markus then did a couple of experiments that you can see here in some bottles here on the right, where they grew this microbial strain, the Synechococcus, in the presence of different concentrations of iron 2, very low concentration, 7.5 micromolar, 30 micromolar, 300, and almost 5 millimolar. And you can see here on the left, the microbes are pretty happy, they grow nicely, you see the green color. And then towards the right, you see that there's obviously some activity. They also produce some oxygen because there's some oxidation, but the cultures definitely don't look very happy anymore, already suggesting that there is an increasing harmful effect of the iron 2 on these microbes. We then determined or quantified the growth rate, again, depending on the iron concentration. And you can see here a significant decrease of the growth rate in these experiments that go from 7.5 micromolar up to 5 millimolar iron 2 concentration. Again, suggesting that iron 2 is toxic to these microbes with increasing concentrations. And when thinking about toxicity of iron in the presence of oxygen and light, we always think about this kind of radical chemistry that you can see here at the bottom, because it's known that oxygen reacts with iron 2 and it forms all these radicals that you can see here highlighted in color. And we then quantified these reactive oxygen species in setups with different concentrations of iron 2. And you can see here an up to 5-fold increase in concentrations of these radicals, suggesting that these radicals play a role in the toxic effect of the iron 2 on the microbes. We then also determined the actinic yield, yeah, that means the efficiency, how these organisms use the light energy. And again, you see that with increasing concentrations of iron 2, we see a decreased actinic yield. That means a decreased capacity to produce oxygen. And this is also what we saw in these bottles that I just showed you before. Now, what can we do with this kind of data? We then worked together with a modeling person who helped us to put together all the equations for the oxidation rates, for the upwelling, and for all the processes that are relevant in such a model basin of a few hundred meters depth. And we then considered a photosynthesis inhibition parameter that we got from the data that we collected in the laboratory with this microbial strain that I just showed you. And what we concluded at the end is that it looks like iron 2 is indeed toxic for oxygenic photosynthesis because of the presence of these radicals. And this could imply that the timing of oxygenation of the atmosphere is controlled by submarine volcanism, because this controls how much iron 2 is then brought into the oceans. And if there's more iron, then the microbial activity is decreased. And if there's less iron, the microbes, they grow up and produce more oxygen. And then if there's a pulse of iron again, there's oxidation. Now, this was what we assumed at that time, until recently, one of my current PhD students, Caroline Dreher, repeated some of these experiments, and she made the system a little bit more complex, and she added silicate. And you can see here two experiments that she did. On the left, 2.5 millimolar iron 2. We also grew the cyanobacterial strain PCC 7002, and she observed the activity of these microbes regarding the oxidation of iron. That means the abiotic oxidation. They produce oxygen, and the oxygen reacts then with the iron 2 and produces these orange iron 3 minerals. If the microbes are happy, you would also see a significant increase in cell number, first indicated by the numbers that we counted here, and we saw an increase from 7 times 10 to the 6 cells per milliliter to 2 times 10 to the 7, but we did not see this characteristic nice green color. However, when she added 2 millimolar silicate, you can see the results on the right. We see that the microbes happily grow. There's also iron oxidation, and we see a significantly higher increase in cell number, up to 7 times 10 to the 7 cells per milliliter. We then also quantified it, and I would like to show you the data here on the left for the experiment without silicate. What you see here is over time, over 25 days. On the left, the ratio of iron 2 to iron total. That means if there's only iron 2 in the beginning, we have a high ratio of almost 1. And here we see oxidation, and we see that after 20 days, we have oxidized all of the iron, and there's a little bit of oxygen accumulating, up to almost 70 micromolar. And here on the right, you can see what happens when there is silicate present. In this case, we don't oxidize all of the iron, because we saw that some of the silicate aggregated with the iron and formed complexes. Obviously, this lowers the toxicity of the iron 2. That means we don't oxidize all of the iron, but also there is less toxic effect. And this leads to an accumulation of, in this case, 350 micromolar of oxygen. That means it suggests that the silicate can actually lower the toxic effect of the iron for the cyanobacteria. And this is something that we are currently continuing to look into, and as part of Caroline Dreher's PhD thesis. Okay, this was the first part that I wanted to show you. In the second one, I would like to show you the potential that anoxygenic photosynthetic iron oxidizing bacteria have for the deposition of banded iron formations. And these organisms, they have a really remarkable metabolism, because they can simply grow in the presence of light with iron 2. You can see that here in these two bottles. On the left, you have a clear aqueous solution of iron 2 with some vitamins and trace metals. Then we add some of these iron oxidizing bacteria, wait 5 to 20 days, and they produce these orange iron 3 oxyhydroxides that you can see here. Here on this electron micrograph, you can see one example of such a strain, Rhodobacter ferrooxidans, strain is W2. You can see the cells, and you can also see some of these mineral particles that they are producing. And they produce iron 3 oxides in the absence of oxygen. And here on top, you can see the equation for the reaction that they catalyze. Now, what we did over several years was we grew these microbes in the lab and did ecophysiological studies to determine how much iron can they precipitate under different conditions, considering temperature, pH, water depth that controls the light intensity, also the presence of silicate, and so on. That means we simulated the ancient ocean in the laboratory. We did that in some bottles, but also initially we had this reactor set up here where we studied the precipitation of the minerals and collected some particles. Now, I don't want to show you all of this data because it's many different plots of rates under different conditions. But what I would like to show you is the final result. And the final result was that considering different strains and all these parameters, we estimated that a layer of about 20-meter thickness of microorganisms at a reasonable cell number in an ancient ocean can oxidize iron and produce or precipitate about 2 millimeters of hematite per year. And this is the number that we can use and compare it to real BIFs. And when I asked my colleague Kurt Kronhaus whether this is realistic or not, he told me that for the Hammersley BIF, you need about 1 millimeter hematite per year. Now, what that means is that these microorganisms, such a layer of photosynthetic organisms, they could have precipitated enough minerals to explain the presence of these rocks. It doesn't mean that they have done it, but it shows that they have the capacity, the oxidation rates, and the metabolism under all these conditions to catalyze the reaction and to produce enough minerals. And we considered, as I mentioned, different factors like pH, temperature, also iron 2 concentration, the simultaneous presence of hydrogen, also sedimentation rates. This was always one concern. Are the microbes removed too fast together with these particles? But we showed that actually a reasonable number of microbes always stays in the water column to maintain this metabolism. Okay, now the third part of my talk is about the banding that we see, this characteristic alternating banding of silicate and iron minerals, and about the potential role of bacteria in achieving such a banding. Now there are different kinds of bands. There are very small ones, very thin ones, even on the nanometer scale, on the low millimeter scale, tens of millimeters, and even larger ones. And different mechanisms have been suggested to be responsible for this layering, for the formation of these different bands. And I just list a few of these potential explanations here. It was suggested that evaporation plays a role, changes in ionic strengths, maybe episodic hydrothermal activity, some surface currents, upwelling, but also compression of iron silica gels and separation of iron and silica. Now, when I'm thinking about microorganisms, I'm thinking about controlling their activity potentially by temperature. And this, of course, leads to the questions, did seasonality play a role? And then when looking into the literature, you actually see that already many decades ago, people have suggested that annual seasons seem to be the only modern climatic rhythm of sufficient contrast, and so on, to explain this alternating precipitation. And even in the Klein review 2005, it has also been mentioned again. That means we thought about whether such a temperature change of a slightly higher to a slightly lower and slightly higher temperature could initiate alternating precipitation of minerals. And a couple of years ago, one of my first PhD students, Nicole Post, she did nice work where she quantified iron oxidation rates by different strains. And you see here at the bottom the names of the 3 different strains. We chose these different ones because they are purple sulfur, purple non-sulfur, and green sulfur bacteria. We have representatives of each of these groups of anoxygenic phototrophs. And what she then did is she determined the oxidation rates. And you can see that here in this plot here, from the left to the right, the oxidation rates, these different symbols, the triangles, the circles, and the squares, are actual data that give you values, and it doesn't give you the absolute values, but relative values, how the oxidation rates increase when we have 25 degrees. And when we go towards lower temperature, you see that the oxidation rate decreases. And here you see what the consequence for the iron precipitation would be. When they are very active, they will produce a lot of minerals. When they are less active, of course, they will produce less iron minerals. Now, the precipitation of silica behaves exactly the other way around. When the temperature is high, the silica stays in solution. but when the temperature is dropping, you would expect the silica particles to precipitate. And this could potentially lead to such an alternating precipitate of iron, iron silicate, and then iron again. Now at this time, Nicole based this mainly on the oxidation rates and on some modeling data. Yeah, she didn't do real experiments. This was done recently by one of my PhD students, Manuel Chart, who is now a postdoc in Canada, who actually did such experiments by using a marine phototrophic iron oxidizer, Photovolum iodosum. He used concentrations of a few millimolar of iron 2 and silicate, and he exposed these reactors first to higher temperatures and then to lower temperatures. And of course, the 4 degrees is not realistic for an ancient ocean, but what we wanted to show is just the effect of the high to the low to the high to the low temperature. And I would like to show you what kind of results you got. Now, on this plot, you can see here at the bottom, you can see the time in days. And here in colors, you can see the warm in brown and the cold in blue periods. Here, the arrows indicate when Manuel respiked the iron and the silicate. Why is this necessary? Because it's not a continuous inflow of iron and silicate. We always spiked it. And then you can see here the dissolved iron concentrations in brown and the dissolved monomeric silica concentrations in blue. Now, what you see here is when it's warm and the microbes are there, they are active, they always consume the iron 2 relatively quickly within a few days and bring the concentrations of iron 2 down to almost 0, precipitating iron 3 minerals. You also see that in the cold periods, the silicate is immediately precipitated and there's an equilibrium concentration that is reached. But again, it's cold and we precipitate silicate minerals as expected. Now, what we also see that is during this phase of iron oxidation, surprisingly, we maintain our silicate at relatively high concentrations. And that was a big surprise to us because we expected when you form these positively charged iron minerals, the silicate will stick to it and will precipitate out together with the minerals. But then we looked for an explanation, and what we found is that the biogenic iron minerals are actually not positively charged, but they are negatively charged because of all the organic molecules that are associated with the minerals. These organic molecules come from the biomass from the microbes. And this suggests that the negatively charged iron minerals can repulse or can reject the silicate, and therefore the silicate stays in solution. And this could explain why we can precipitate the iron without the silicate, and later we can precipitate the silicate without the iron. Now, we did some abiotic controls, of course, where you see that the iron stays in solution, when there are no microbes present. And you also see that, of course, when you have no microbes present, when you have the iron and the silicate, you produce some co-precipitates of iron and silicates. How do these precipitates look like? Here, this is an example of such sediments that Manuel put together to illustrate how these alternating layers look like. Here on the left, you can see how the alternating iron and silicate layers look like before compaction, and then he centrifuged them. And this is the result, what you can see here on the right. And when you compare that to one of the BIF thin sections that I collected once in South Africa, I think there are some similarities, suggesting that maybe such an alternating layer, you see the layers are not always uniform and equal. They, depending on how the mineral sediment in the currents, they vary a little bit in thickness. But this suggests to me that such an alternating temperature cycle could actually explain the alternating deposition of iron and silicate minerals. And the band thickness varies with iron 2 and silica input and with the lengths of the different periods. Okay, now in the last part of my presentation, I would like to talk about the following processes after deposition of the minerals. And as I mentioned before, after oxidation, we have the precipitation of these particles. We have a whole series of microbial processes. This is something I will not talk about today. This was covered recently by a paper that was published by Manuel Chart, who did his PhD in my group, where he studied the combination of iron oxidation by phototrophs and the iron reduction by an iron reducing bacterium, and showed what happens when you have several of these cycles, what kind of minerals do you precipitate. One of my current PhD students, Caroline Dreher, she actually now goes the next step and combines cyanobacteria with iron reducing bacteria, again studying What happens? What kind of minerals do you produce when first cyanobacteria lead to iron oxidation and then an iron reducer gets active? And then again, you have a cyanobacteria producing oxygen. That means you have iron cycling happening in such a system. But what I would like to do now is to show you what happens when these minerals that are produced, that are even remaining after the microbes have been active, and the biomass, if they react together at higher pressure and temperatures, leading to abiotic iron-3 reduction. That means we're interested in how the electrons from the organic carbon are used under these conditions, abiotically, to reduce some of the iron-3 back to iron-2. And of course, there will be some oxidized form of carbon remaining. And what can happen there? Potentially, you could form minerals such as siderite and magnetite, that means an iron-2 carbonate, or a mixed valent iron-2, iron-3 oxide, magnetite. There's the potential for forming oxidized gases like CO2 or carbon monoxide, also kerogen. Now, how do we study this? How do we approach this? And what kind of questions do we ask here? The first one is, what is the fate of the organic carbon? Yeah, what happens to this biomass when these cell mineral aggregates containing the biomass are deposited? That means you have the biomass plus the iron-3 minerals. Are there gases formed, such as carbon monoxide, carbon dioxide, or even methane? And this would be relevant because you know in many of these banded iron formations, we don't find a lot of organic carbon. And this reaction and the release of these gases into the atmosphere could explain where the electrons from the biomass are nowadays. They are just gone from the sediments because they are transformed into methane. Then the second question is, what is the efficiency of these reactions, of this reduction of the iron-3 minerals using the electrons from the biomass in such an abiotic reaction? That means, what is the extent of iron-2 formation and what is the identity of these iron-2 and iron-3 minerals that are produced? Now, how do we approach this? What we did is we used these gold capsules that you can see here at the bottom, and we filled them with different materials. We used hydride and polycrystalline iron-3 hydroxide as an example of the mineral produced by the bacteria, or we used a more stable, more crystalline iron-3 oxide hematite. And then we used 3 different types of biomass, either glucose as an easily degradable source of carbon. Then we use microbial biomass, where we just grew some other bacteria and collected the biomass. And either we left it as it is, this is the so-called untreated biomass, UBM, or we pre-treated it at high temperature to have treated biomass. Yeah? And the idea was behind that this simulates this second step of transformation of the biomass. Then we also use different ratios of biomass to iron. What is the motivation for that? When you have phototrophic iron oxidizing bacteria, they use the electrons from iron-2 to fix CO2 into biomass. And there's a clear stoichiometry. That means you need 4 electrons to fix 1 CO2. That means the maximum ratio of iron to carbon that you can have is 1:1, yeah, regarding the electrons. But because we know that some of the biomass stays in the water column, is not deposited, we estimated that the real ratio of the electrons in biomass versus iron-3 that can be accepted by the iron-3 is around 0.6. Now the 2.4, it comes from this idea that if you have cyanobacteria, they use water as an electron donor, and they can produce as much biomass as they want. And only part of the electrons then indirectly, via the oxygen and so on, are used to oxidize the iron-2. That means you could have an excess, you could have an excess of electrons from the biomass. And this is where the ratio 2.4 comes from. Okay, and then what we do is we take these gold capsules, we incubate them at high temperature and pressure for several days to months. Okay, now I can see this funny line on my screen. I don't know whether you can see it as well.
Yeah, I'm trying to clear it. Sorry
Okay.
about that. I don't know how it happened.
Okay, no problem. I cannot remove it.
It'll be clear in a second.
Okay, I will just continue, And then we have this high pressure autoclave that you can see here, where we have 3 reactors where we can put in the capsules and then incubate them at high pressure and temperature. What we can do is with these capsules, we can put them in such a little container, close it on both sides and poke into it, and then we can quantify and identify the gases that are produced during the reaction. You can see it here from the side. We then have a syringe, yeah, to extract the gases and to make a hole into the capsule. And this is how we can extract the gases. And then we inject the sample into a GCMS and analyze the gases. Now, what did we find? When we used glucose as an electron donor and reacted it with these iron 3 minerals, yeah, we saw carbon monoxide, we saw carbon dioxide, but we did not see any methane. Overall, within a few weeks of incubation, we saw up to 8% of the carbon present in carbon dioxide and 10% in carbon monoxide. This is quite a lot considering that it was incubated for only a few days. Now, we also observed some compounds like furane and acetone. And this got, when you look into more details, you can see that actually at low concentrations, a whole series of molecules that are volatile are produced under such a simple scenario. Yeah, just take the organic carbon, the glucose, yeah, mix it with iron 3 minerals, and this is the product. Now, when we used biomass, where we have, of course, proteins, where we have all the sulfur, nitrogen, and so on, we saw more compounds. We found sulfur-containing hydrocarbons, dimethyl sulfide, for example, methanethiol, and so on. And what does that mean? That means when we treat such materials, such biomass at high temperature and pressure, we lose reducing equivalents. That means we lose carbon from the sediments. And this could potentially, to some extent, explain why we don't find a lot of carbon in the banded iron formations, because the biomass is simply transformed over time into volatiles that escape. Now, how about the minerals? How did we look into the mineralogy? We used micro X-ray diffraction. You can see the machine here, and we used Mössbauer spectroscopy. Mössbauer spectroscopy is an iron-specific technique where gamma radiation is emitted from a radioactive cobalt source and specifically absorbed by iron 57. And we can use that to quantify iron 2 and iron 3 and also to identify the minerals. And I will show you some of these spectra. Now here, one of the initial experiments where we used ferric hydride, treated biomass, that means the more stable biomass, at a ratio of 2.4, simulating an experiment where we had cyanobacteria producing excess biomass. And this spectrum was measured at 140 Kelvin. Now what you can see here is this typical sextet, and you can see this doublet in blue, the sextet in red. And based on references that we synthesize and analyze, I can tell you that this shows the formation of hematite and some siderite. That means some of the ferric hydride, this poorly crystalline mineral, got transformed into a crystalline phase, and some of the iron 3 got reduced to iron 2, forming siderite. We saw in this case that up to 20% of iron 2 was formed already within 20 days. We only saw siderite and hematite, but no magnetite, and that's very important. Now we did a ton of these different experiments, and I would just like to show you some here. Again, we have ferric hydride plus treated biomass at a low ratio, ferric hydride plus treated biomass at a high ratio, then the untreated biomass at both ratios. And here you can see the reference data for micro-XRD, the diffraction pattern for hematite in red, for siderite in blue, and for magnetite in green. And what you can see here is that, again, under all these conditions, we only produce siderite and hematite, but no magnetite. And this is interesting because we see in many BIFs magnetite, or in some BIFs, and the question is, what is the origin? Is this formed by iron-reducing bacteria, or is it formed by this abiotic reduction of iron 3? And here even more experiments where we did not start with ferric hydride, but with hematite. And you can see whatever biomass we used, glucose, treated or untreated biomass, whatever ratio, we always see siderite and hematite. Yeah, when you look at the reference patterns here, we don't see signals here where there are the green lines. That means there was no magnetite produced, only siderite and hematite. And this suggests to me that the magnetite that we see in BIFs is probably formed by microbial iron reduction. And this is also supported by iron isotope studies. Clark Johnson did many of them, and other researchers as well, suggesting that magnetite is of biogenic origin. Okay, at this point, I would like to summarize what I tried to show you. First, in the first part of the talk, I showed you that iron 2 could have been toxic for cyanobacteria. It could have delayed the oxygenation of the atmosphere. However, recent experiments showed that this toxic effect is potentially rescued by the presence of silicate, by forming these iron 2 silicate aggregates that are not toxic anymore. Then in the second part, I showed you that seasonal temperature cycles can actually cause such an alternating banding of iron and silicate minerals by controlling microbial iron oxidation. And then finally, I showed you that when we take these biomass iron 3 mineral aggregates and incubate them at higher pressure and temperature, we find the minerals, or we produce the minerals that we find in BIFs, like siderite and also hematite, but we don't find magnetite, suggesting that the magnetite is produced by microbial iron reduction. And at the end, I would like to thank, of course, my group and the students in my group who did the work. Kurt Kronhauser, who is a long-term collaborator, Betsy Swanner, who is now in the US, and the German Research Foundation for their funding. And I would like to thank you for your attention, and I'm happy to take any questions.
Thanks, Andreas. That was really good.
Thank you.
We're going to go ahead and open up for questions, so you can raise your hand to ask it yourself or type it in the chat. I have one that was asked earlier relating to the difference between lab experiments and water depth in an ocean. In the simulation for the stratified cyanobacteria using glass beads, how do you account for the ambient pressure as a result of weight of water sitting above the bacteria? And do the free radical reactions also follow similar chemical kinetics underwater when compared to the same reaction occurring up here in a terrestrial environment?
Okay, good. Regard the first question, we did not account for any pressure effects. Yeah, and I mean, if these microbes live in the photic zone at the surface, I think the pressure difference is not changing the activity too much. Therefore, I don't think that this is necessary. However, what this also means, the setup that we used with the glass beads is we did not account for any sedimentation. And this is, of course, a huge limitation, yeah, because when you have these minerals produced, they also react as catalytic surfaces. And we cannot exclude that this also played a role, yeah. And therefore, we are actually currently planning experiments in long columns, not only these very short ones, 20 to 30 centimeters, but we use long columns where we can have stratification of maybe the cyanobacteria and the phototrophics. We have a light source coming from the top, and we can actually also take into account the sedimentation. And the second question was regarding the role of the radicals. I mean, these reactions that I showed you, the equations of the different radicals with iron, of course, they depend on many different environmental factors. Yeah, the salinity, the pH values, the presence of organic matter is a very important one. Yeah, because these radicals will definitely react with natural organic matter, whatever that is, in a terrestrial environment, but also in an aquatic one. If there are organics around with ketones, yeah, with maybe other metal ions like manganese, they will react with these radicals, and this will lower the toxicity of the radicals for sure. I think this is an important part of what has to be done, that we make the system more and more complex, include silicate, include relevant organic matter, and see whether this toxicity effect still plays a role or not. Just based on the silicate experiments, I already believe that the toxic effect of the radicals is lower than we assumed before.
All right. I have a question that might cascade a bit. So the concept of oxidizing iron for microbes, I mean, they're going to use iron, they also need to get oxygen in some way. So how are they doing this? And is it with ambient oxygen or a complete absence of oxygen, and they're getting it somehow in some other way?
You mean the microaerophilic iron oxidizers, yeah? The ones that
Yeah.
need, I mean, they are the anoxygenic photosynthetic ones. They don't need oxygen at all. The oxygen that is present in the minerals after anoxygenic oxidation comes from water, yeah? They oxidize iron 2 to iron 3, and then the iron 3 ion is hydrolyzed by hydroxyl ions from the water. That means the oxygen doesn't come from O2. Now for the microaerophilic iron oxidizers, the ones that use oxygen, yeah, go down here, these ones here at the bottom, they use molecular oxygen to oxidize the iron 2. Now what is really interesting is where do we find them nowadays in the environment? We find them in scenarios where we have gradients of oxygen from the top and gradients of iron 2 from the bottom. They live at the interface. They like to have low iron, low oxygen concentrations because this limits, again, the formation of toxic compounds, and it also limits the competition with the chemical reaction. Because if we try to cultivate them with high iron and high oxygen, the chemical reaction is so fast that the chemical reaction outcompetes the microbes. Now, in such an ancient ocean system, you can imagine such gradients because the microbes, the cyanobacteria, produce oxygen that is present in the photic zone on top. And then the iron 2 comes from bottom. You already established these gradients. And when you go into a modern environment, we actually see these gradients everywhere. This is where these microaerophilic iron oxidizers have been isolated from. Hydrothermal vents, yeah, all kinds of iron mats that you see in the environment. It's always in such gradient situations where you have some oxygen and a little bit of iron. And this is where they like to live.
Okay, so for microaerophiles, then it seems like you need a significant amount of oxygen for in the Earth system, surface system, to really give them what they need. I mean, I guess a good question is, I mean, each of these fall under the larger category of zeta proteobacteria. Is that correct?
Yes.
So do we have
some
of them do? Do we have much I guess molecular clock information on when these different iron oxidizers would have split?
I mean, that is a good question. I think that, I mean, you would imagine that the anoxygenic photosynthetic ones are coming earlier, yeah? And I don't know whether there are molecular clock approaches for the microaerophiles. I actually don't know that. But one interesting point that you made is regarding the oxygen concentration. We know that they have their optimum growth in oxygen concentrations of around 20, 30 to 40 micromolar. That's relatively low. I mean, they can grow at even lower oxygen concentrations. That means we really don't need a lot. It's not, we don't talk about 200 micromolar, yeah? It's a factor of 10 less. And you can imagine, they will also grow at 10 micromolar or 5 micromolar, but then they are just slower. And therefore, low concentration of oxygen are, I would say, relatively realistic in such a system.
Do they then outcompete the anoxygenic ones at that point?
I mean, it's a good question. What do you see in a modern sediment? You see the typical stratification, yeah, of cyanobacteria. Yeah, you have then fermenting bacteria, and then you have different layers of anoxygenic photosynthetic ones. And this is what you see in modern lakes. There are a few lakes that show this stratification, yeah, where you have an anoxic layer with iron 2, and you have an oxic layer at the surface. That means they compete for light, yeah. But of course, the anoxygenic phototrophs, they go down and they can live with lower light intensities. Especially the green sulfur bacteria are specialized to conditions with low light energy. This is why you usually find them underneath the other ones. That means there's a stratification by using different pigments, yeah, by adapting to different concentrations of oxygen. Yeah, oxygen sensitivity is something important. And in one of our publications, we actually calculated how much iron the anoxygenic phototrophs could oxidize when there's upwelling. And it suggested that they can oxidize when you have a layer of 10 to 20 meters of these microbes, what is realistic compared to the Black Sea for the sulfide oxidizers. If you have such a layer, they can actually oxidize all of the iron 2 that comes from the bottom before it even reaches the layer where we have the cyanobacteria. I mean, microbes, they compete in the environment, yeah, and they find strategies, yeah, to get their substrates as quickly and as efficiently as possible. And I mean, the cyanos, they need iron only as a nutrient, yeah, but they don't need it for a living, yeah. And therefore, the anoxygenic photosynthetic ones, these will be the ones oxidizing the iron. And of course, they compete with the microaerophiles, if there are microaerophiles at the same time. But this is now when you go into a modern environment, you find microaerophiles in environments, and there can be anoxygenic phototrophs at the same time, yeah, when you have light energy there.
Okay, great. I'll hit you with one more. In ferruginous conditions in pre-GOE settings, you might expect that sort of the bioavailable nutrient situation to be different than post-oxygenation of the oceans. So could you maybe comment on how that might be different and if it may be contributing to the delayed rise of cyanobacteria oxygen production in preference of these iron oxidizers?
Yeah, absolutely. I mean, phosphate is the nutrient, yeah, you always think about first. And what is interesting, I mean, when you take an iron 3 mineral, positively charged, when you add phosphate, there is significant and large removal of phosphate. This removal changes already when you have silicate present, because it competes with the silicate, yeah, for binding sites. And of course, it competes also with biomass. That means in a scenario where you have biogenic minerals, there's actually less phosphate bound to these biogenic precipitates. We know that already for the precipitates produced by anoxygenic photosynthetic ones. For the cyanobacteria, this is something that Caroline Dreher in my lab is actually studying in the next phase of her PhD. She's now combining these cycling experiments with cyanos and iron reducers with phosphate and nickel amendment. And she studies the fate of phosphate and nickel. That will tell us something about how these precipitates will then influence the nutrient availability for cyanos. What is also interesting, when we grow these bacteria, it's really surprising how low we can go with these organisms. Yeah, it's just by accident. Caroline Dreher showed me yesterday a photo, yeah, where she cultivated these organisms at different phosphate concentration, and you see a clear dependence. They like phosphate, yeah, they need the phosphate, but they can also be active at lower concentrations, and it looks like they efficiently recycle it. And in many cases, when you give them high concentrations, they also store it in the cells. And then when you lower it, they are very efficient in recycling. And maybe the cells get a little bit smaller, maybe less proteins are produced, but they find ways to still survive even at lower concentrations.
Okay, thanks. I actually just got another one sent to me. Sorry, the previous question from the chat was K-Sundersar1, and he just asked again, saying, when exactly did this niche partitioning for oxidizing substrates arise? And what were the major driving forces for the partitions? And as Alex said previously, would this partitioning correspond to their time of phylogenetic divergence?
Mm-hmm. The question of the phylogenetic divergence, I cannot answer. But the first part, I mean, what would you expect? In an ancient ocean where you had no oxygen, only very little oxygen, this was a paradise for the anoxygenic photosynthetic iron oxidizers, yeah, because they had unlimited space, yeah, they had a ton of iron 2 light energy. And as soon as the cyanobacteria evolve and you produce some oxygen and you have oxygen at the surface, these microbes, yeah, will not oxidize iron 2 photosynthetically anymore, yeah, because they cannot do that in the presence of oxygen. Interestingly, some of them, they can also grow with oxygen, yeah, but then they don't oxidize iron. They can maybe oxidize organic substrates. And whether this was possible at the time already, we don't know. But I would expect that then the cyanobacteria evolve, produce some oxygen, and then this pushes down the anoxygenic phototrophs, yeah, so that they find a layer where there is still iron 2 and no oxygen. And then the microaerophilic iron oxidizers can potentially be active in the surface layer. Now, what is also interesting is all the modern microaerophilic iron oxidizers, as far as I know, they have been isolated from sediment, from hydrothermal vents, but from solids. I don't think that some of them have been really isolated from a water column or from a lake water column. Yeah, I mean, there's no ocean that can be used for this kind of study, but maybe one of these model lakes, Lake Perrin, Lake La Cruz, I don't know whether someone looked for microaerophilic iron oxidizers in such a water column, because then you can study how fast they expand. Maybe it changes over the year, yeah, when their iron gradients change, or even the temperature. So, but it's a good question, yeah. That when did this separate? I mean, I think it separated when cyanobacteria started to produce enough oxygen.
Okay, great. We don't have any hands raised or more questions.
So maybe I can ask. Great talk, Andreas. So
Thank you.
Wondering if you could comment was another paper on experiments done, I think maybe in your lab. I forgot my first author, maybe Herman or somebody, on toxic effect of iron, and they had very different conclusions. So maybe you can expand, like, did they include silica in their experiment, or why they derived two different conclusions? And the second part of my question, do we know about a toxicity effect of iron on anoxygenic iron oxidizing bacteria? Does it have any role?
Yeah. Okay, the second question is a very good one. But we also see that for the anoxygenic phototrophs, and by the way, also, for example, for nitrate-depending iron oxidizing strains, some of them, they have a clear upper limit until they like the iron 2. And for some of them, it's actually really sharp. Yeah, you go to 4 millimolar, they love it, and you go to 6 millimolar, they are okay, and at 8 millimolar, they don't oxidize anymore. That means there is also, even in the absence of oxygen, yeah, there seems to be a toxic effect. Some of these microbes, they react by excreting EPS, extracellular polymeric substances, organic compounds, and we believe it's because then these organic molecules bind the iron and help to detoxify. It's basically a protection mechanism. It could also be that the EPS, and we see that the EPS together with the cells and the iron minerals that are produced, they form aggregates. And you can imagine that then you have diffusion of iron 2 into these aggregates, and maybe the cells inside the aggregates, they are exposed to lower concentrations. That means it looks like these microbes themselves already react to the high iron concentrations and find ways to detoxify by protecting themselves. Yeah, that's the, I think, regarding the toxicity for the anoxygenic phototrophs. Yes, I agree there. They are also sensitive to iron. Iron in the presence of light, yeah, and organics is always reactive, yeah, because the iron takes electrons, yeah, from the organic matter. Then you form also organic radicals, and these organic radicals can also react with the cells. This is always stressful, and we see that there are even in the absence of light, yeah, when you have organic matter reacting with iron, you form these kind of reactive species. And then regarding the effects of iron 2, the toxicity effects observed in different systems, I think that it's really important that people consider different parameters. And there are a couple of studies, yeah, and I mean, there was some done where only iron 2 and growth medium and microbes were used. Now we added the silicate, some other people added maybe organic matter already. And this is what we need to do. We need to consider all these different factors, pH values, salt concentrations, different types of organic matter, terrestrial versus marine, and just see how this influences the formation of these radicals. And I think this explains why in these different studies we just see different results. And yeah, I don't know exactly which study you refer to, but I know that there are some that use different conditions. And this is like with the mineral precipitation. Yeah, it depends on what kind of concentration you use. If you have low concentrations and low rates, you produce more crystalline mineral phases. If it's oxidizing fast, you make more polycrystalline ones. Therefore, we need to decide what is most realistic scenario.
Would you expect a different toxicity level for like microaerophilic versus anoxygenic versus cyanus?
Yes, absolutely. Yeah, I mean, definitely for the, I mean, in the presence of cyanose, this is the worst case. You have light and oxygen and iron and organic matter. Yeah, this is the, I would say, the worst case. For the microaerophils, you have no light. Yeah, but you have also oxygen and iron. And then for the anoxygenic phototrophs, you have the iron and the organic matter, but you have no oxygen. Yeah. And I think this, therefore, I would expect it makes a difference just because of the way how these radicals are forming and how they react with the cells. Therefore, I would expect that it's different. But then, of course, there is always strain dependence. Did they find ways to adapt? Do they have they been isolated from an environment where they are exposed to these concentrations, to certain concentrations of radicals?
Thank you.
Yeah, thank you. I saw that Lee also. Right.
Yeah, Lee, go ahead.
Yeah, thanks very much. Real tour de force. Appreciate the talk. I'm wondering, about the seasonal model for the iron and silica banding. Other things vary seasonally, and what I'm thinking about in particular is upwelling rate. And I'm wondering if you've taken the modeling a step further and thinking about supply rates of iron and silica, rates of accumulation on a seasonal basis, maybe upwelling and supply, and then stratification in the basin. That next level of complexity of how the basin might generate that. Is there additional support or complexity there when you think about other factors?
Yeah, it's a very good suggestion. We have not done that, but as I mentioned earlier, we are currently building this large column where we have an inflow of iron and silicate, where we don't need to inject every time. This experimental setup was not ideal, because every time when you inject, you rise the concentration, and then the microbes start consuming it, or it's precipitating, and you inject it again and you rise it up. It's always a pulse. I think this is not ideal. It's much better when you have a constant inflow. And then what we can do is what you suggested. We can play with the inflow rates or the inflow concentrations, and then we can see what is happening. I expect, for example, just because of these toxicity effects, that we will shift the stratification of these different organisms, because if there's more iron, maybe there are more radicals, maybe the zones get more compact where these microbes are active. Maybe they slow down with their activity because of toxic effects. And then, of course, you will also maybe change the ratio of iron to silicate that is precipitating, because it could be that at a certain ratio of iron, if it's increasing, the particles are not exclusively negatively charged. There are also examples where particles form patches of negative charges, positive charges, negative charges, positive charges. And as a consequence, these particles just aggregate and form larger structures that then sediment out quickly. And it could be that a change in such an inflow rate could also significantly influence the sedimentation of the particles, just because this surface charge changes. But these are, I think, excellent experiments that we need to do, or someone.
I look forward to that. Just one other question or comment. Have you looked at metal binding ligand production? I know cyanobacteria produce copper binding ligands, and this has an effect on toxicity. And I don't know if the same thing with siderophores are, so I don't know if you've looked at that in your experiments at all.
We have not done that. One reason is simply that in all these experiments, there is so much iron in there that they would never be iron limited. There is no need for them to produce siderophores. And we did not look into other metal binding ligands. But I think for the siderophores, because we have up to millimolar concentrations of iron, or at least tens or hundreds of micromolar, the iron is definitely not limiting for these organisms.
Yeah, no, I'm thinking more on the detoxification side of things, and maybe siderophores are, that's the wrong example, because the copper binding ligands are detoxifying.
No, that could be. I mean, we see increased EPS production for some of the iron oxidizers, and EPS is also a metal binding ligand. It's just not defined like a siderophore. It's not one compound, but it's a whole matrix of organic molecules that is binding metals and accumulating. And we also see precipitation of minerals at these EPS structures. It looks like some of these photosynthetic iron oxidizers are producing organic structures to deposit the minerals. It's not only these microaerophiles with the twisted stalks and the sheets. It also looks like some of the other iron oxidizers are producing organic molecules to direct the precipitation of the minerals away from the cell.
Cool. All right. Thank you.
Thank you.
I guess in the line of seasonality, I'll just point out that Marguerite Lantink, who gave it, is present here today. Actually, she gave a talk on linking Milankovitch cycles to iron formations on the Kuruman, and she talked about the Kuruman and data from the Brachman iron formations. So that's on YouTube for people to check out if you haven't already. So we've hit a point where we're out of questions in the chat or hands up, but I'll give people a few minutes to step in for discussion, or a few seconds.
I should know it, but how large variation in temperature in modern deep ocean? And did you try to use the same range for your experiments.
Yeah. I mean, in modern deep oceans, yeah, it's not not significant. Yeah. I mean, we talk about the photic zones. And there are oceans where in the photic zones you have temperature variations of 10 to 15 degrees maximum. But of course is a lot. Yeah. I mean, the experiments that we did, we changed the temperature by 20 degrees. But I think based on the modeling, you see that it's already significant that when you change it by 10 degrees. Also, because I mean, the key point is that we separate the iron from the silicate because of the negative charge of the iron. And this gives the silicate time to accumulate and then to reach oversaturation, where I would expect precipitation just when the temperature drops maybe by even only 5 degrees. And the microbes, they are very sensitive regarding their optimum oxidation rates. Yeah, when you drop the temperature by 10 degrees, this has a significant impact already. And we only need to lower the rate by a factor of, let's say, 2 to 5 or so. Yeah, then it's already significantly different regarding the amount of precipitates that we see.
So what would happen if you would have a pulse of iron and silica? So you deliver it, you would precipitate iron, and when as iron is exhausted in water column, would you precipitate silica? Do you need a temperature change for that?
At the end, it's chemistry. Yeah, it's oversaturation. And what is it? If you start precipitating the first minerals, and if you have an oversaturated solution of silicate, it could be that there's also silicate precipitating at the same time. And it could, when you have a lot of iron, I predict that then the iron particles are not homogeneously negatively charged. That means you will have maybe some small areas where there is positive charge, and this is where silica will bind. And then you have a heteropolymer, yeah, consisting of EPS, microbes. There is maybe a silicate nanoparticle here, and you have a big chunk of iron oxide. And then, of course, the question is what happens during sedimentation. We did not talk about that today, but we know that already during sedimentation, a significant part of the biomass can be used by bacteria, yeah, and can be oxidized and can also lead to iron reduction. Also an interesting question. When you start reducing some of the iron, do you then dissolve the silicate back into solution together with some iron? Or do you then maybe even form iron to silicate precipitates that then also precipitate out? I don't think that someone has done these kind of experiments. But this is definitely also relevant for such a scenario. And what you just asked with the pulse, if you have a pulse of iron and silicate, if it's oversaturated, yeah, it will precipitate. Then the question is, what will precipitate first? Yeah, that's the lower activation energy, and maybe nucleation sites play a role. Yeah, we know when we have some iron precipitates already, and we add more iron, and then you start oxidizing it, then everything will accumulate, will aggregate, will sediment out. And we see that also for the silicate. Yeah, you just have some nuclei, and then it starts precipitating.
Okay. I guess,
so Andreas,
yeah.
go ahead.
I guess I was more thinking in terms, so if you start with solution saturated with iron and containing silica, and you precipitate iron first from it, so at some point you're left with high silica in solution. Would it, you're saying you need to change temperature to get it to saturation point? Okay.
I mean, we don't need the temperature to reach saturation, but I mean, what we also see in the lab, even when you reach values that based on equilibrium modeling would predict that you see precipitation, you reach oversaturation. And then there are small effects that then initiate precipitation. And this can be, of course, the addition of some nucleation sites, but it can also be a drop in temperature. And then it suddenly nucleates, and then it precipitates out. And when it's warm, it stays in solution. It's already oversaturated, but you don't see anything. I mean, you scratch at the glass wall, and then suddenly it's precipitating out. Just because you have these small glass particles that then function as nucleation sites. But I think this is, I mean, when you reach saturation, yeah, even microbial surfaces can function as nucleation sites. If you have some microbes, yeah, and at the surface you have maybe some calcium or magnesium binding, and that links to the silicate, and then suddenly you start precipitating. I don't think that it's only the temperature.
Thank you.
Okay. Great. I think it seems we've gotten some really good good discussion and everybody's satisfied. Really great talk, Andreas. Thanks for being here today.
Thank you very much for joining, yeah, and for the excellent questions.
Great. All right, everybody. Take care. See you next week.
Bye.
See you. Bye.